Okay, this is the second video on the chapter on amino acids and proteins. First we covered amino acids, and now we'll talk about peptides, maybe more specifically polypeptides. These are nothing more than a polymer of amino acids, so we can have a number of amino acids linked together. So for example, we could have a few amino acids like leucine and maybe arginine. Right, that would be a dipeptide, two amino acids linked together. A tripeptide, we could have leucine, arginine, and maybe a valine or something like that, three amino acids. An oligopeptide, so you have a dipeptide, tripeptide, tetrapeptide, whether. Oligopeptide isn't specific for any particular number, it's just many amino acids. Right, so an oligopeptide is maybe 10 to 50. So oligopeptides are really kind of the same thing as a polypeptide. And what we're really talking about here when we say peptide, we're not really talking about the individual amino acids. What we're talking about actually is a peptide bond. Now you remember in the chapter on carbohydrates we had acetal bonds, right? And I said, well, that's really the same thing as a glycosidic bond. A glycosidic bond is an acetal linkage between two different carbohydrate units. A peptide bond is really an amide bond between two different amino acids. So here we see a dipeptide, we have residue 1. Let's do something like this. So we have an amino acid, alpha carbon, it's R group, and its amino group. What we have is the carboxy terminus, I should say, the carboxylate of one amino acid reacting with the ammonium part, or the amine part, of a second amino acid. Right? So we'll have here our two parts react together. You have the acid and the amine. Remember that chapter on amines and amides? The way we make an amide, we react carboxylic acid with an amine, and we get the amide. So here again we have, this is that second amino acid here in blue, right? The carboxylate, it's alpha carbon, right? So here's its alpha carbon with the R group, the side chain, and its amino group. And then we have this guy on the left, again, the carboxylate, it's alpha amino acid, it's alpha carbon and its amine. Between two different amino acids, we have one peptide bond, right? So here's the peptide bond, and it's really what it is an amide. See the same thing here. Here's a peptide bond between two different amino acids. Here's another peptide bond between two different amino acids. So we have a tripeptide here. The first R1 here goes from the amino group through its alpha carbon to the carboxyl group. Second amino acid from its amino group to its alpha carbon to its carboxyl group. Now, of course, now it's really a carbonyl. Carboxylate or carboxyl group has been reacted with the ammonia here, the ammonium of a second amino acid. So you don't see a carboxyl, but this carbon here was that acid or carboxyl carbon. And then lastly, we have one more amino acid here, right? Its amino group, its alpha carbon, and its carboxylate. Between two different amino acids, we get one peptide bond, one amide bond. Between these two, we have an amide bond, right? Put it outlined here, bracketed in black, and a second one here. All right, those amide bonds are peptide bonds because they're between individual amino acids. In this figure, again, they're showing a stretch of a polypeptide. Right, poly means more than one. We have more than one peptide bond here. So this is a polypeptide bond, and they've kind of just a squiggly here. They kind of broke this off, but we'd have a peptide here. Another, we'll call that R0, right? R0, R1, R2, R3. So these are the peptide bonds, and what I want you to focus on here is if you're trying to pick out where the side chains are, and obviously it's going to get more complicated when we actually start putting in side chains, but what I want you to do is focus on these carbonyls that are kind of sticking out. Okay, so focus on the carbonyls, these carbon double bonded oxygens. I think those are the ones that are easiest to pick out. And except for a couple different amino acids, we had asparagine and glutamine, there's no other carbonyls in any of the amino acid side chains. So they're pretty easy to pick out. So if this is a carbonyl, the carbon next to it, this is its alpha carbon. Right? So this is one amino acid. This carbonyl was a carboxylate. The carbon next to it is the alpha carbon. Going the other way, then is this nitrogen is a different amino acid, right? So here's a different amino acid. Here's its carbonyl. Here's its alpha carbon, right? So looking at this carbonyl, go to the left, you get to a carbon. That's the alpha carbon. These two guys are part of the same amino acid. Go to the right, we get to a nitrogen. This is the amide bond. This is a different amino acid residue now. And by the way, we call the R doesn't really stand for residue, but you can think of it that way. We refer to the various different amino acids in a polypeptide as residues. Residue 3 or amino acid 3. So all right. So here's a carbonyl. You go to the right. That's a nitrogen. Okay, so we know that's a different residue. Here's its alpha carbon. Here's its carbonyl. We look at this carbonyl, you go to the left, we get a carbon, so that's its alpha carbon. You go to the right, a nitrogen, that's a different amino acid, so this would be amino acid 4, residue 4. Now they make some point of talking about the geometry here. I'm not going to focus on that. What I do want you to notice, and remember all these amino acids are chiral, except of course for glycine where the side chain here would be a hydrogen. But notice the side chains kind of alternate. This is going away from us and it's down. This is coming out towards us and it's up. This one's going away from us and it's down. You'll see this quite a bit. So I just want you to notice that the side chains alternate. Right, so we have the alpha carbon backbone here. All right, so here's our alpha carbon backbone. And then the side chains alternate. One's going away and down, one's coming up towards us, one's going away and down. So they kind of go back and forth that way. You'll see this again, start talking about more complicated structures. So as I mentioned before, we have the carboxylate of one. This is a carboxylate of one amino acid reacting with the ammonium of another. And we get the peptide bond, right? The peptide bond between these. Now notice, say we're spelling a word, right? So say we're going to put two letters together, and you're learning the English language or something like that, and you learn that N-O spells no, right? We have an N and an O. Well, we read left to right, and so we could have the same exact letters, right? O and N does not spell no, right? This is a different word. Well, that's what they're pointing out here. So this amino acid, here's its side chain. Here's its alpha carbon. This amino acid, they've color-coded these. This first one is in blue. This is alanine. And this is serine. Serine has the polar side chain with the alcohol, right? It's like a methanol as a side chain. Here's its alpha carbon and its side chain. So we have a dipeptide here of alanine and serine. If we flip this around, it's still the same two amino acids, serine and alanine, but they're isomers of one another, but they're not the same molecule. Okay. Notice the serine here has a free carboxylate, and the alanine has the ammonium. For dipeptide B over here, it's just reversed. It's the alanine that has a free carboxylate, and the serine that has the free ammonium. So the order matters. Okay, so we call the free carboxylate the C terminal, or sometimes referred to as a carboxy, the C terminal or carboxy terminal, or terminus. Sometimes you'll see the word terminus. Okay, and the N here, the N terminal or amino, because this is an amine here, the amino terminus. So these words, amino terminus or N terminal, C terminal, could C for carbon, or carboxy terminal, terminus, same thing. So normally we read polypeptides just like we read these words left to right, right? So NO is not the same, right? Is not equal to, not the same as ON, right? We read left to right. So when we read these polypeptides, we read from the N end to the C end, from amino terminus to C terminus. Now, it helps that this is also alphabetical. A comes before C. So when you're looking at a series of amino acids, and we could, like I said before, we had maybe like leucine, R is arginine, maybe glycine and glycine, right? This would, by convention, this would be the amino terminus, and this would be the C terminus. That would be different from glycine, glycine, arginine, leucine. Here they're asking, determine the amino acids present in each. And you might come across this. They might ask you to do something like this in the homework. I wouldn't ask you to do it. Well, you might see this on the test, because honestly the tests are online, and you do have access to your textbook. So what you really need to do, determine the amino acids present, is, again, focus on the carbonyls. We have two carbonyls sticking out. That means it's a dipeptide. Right? So for starters, we know it's a dipeptide. It says here polypeptide, but this is a dipeptide. Dipeptide, right? Two different carbonyls sticking out. So this carbon, here's its alpha carbon. For this carbonyl, here's its alpha carbon. Of course, the side chain is attached to the alpha carbon. So here, this side chain is just a methyl group. This is alanine. This one's a little bit, the way they've drawn it is a kind of a little bit odd. Let me go ahead and just draw. Here's our alpha carbon with its hydrogen. We have a CH, and then there's a CH3 here, and then a CH2, CH3. CH2, CH3. Okay, so if you look in that table of all 20, you'll see this is isoleucine. Okay. So we have alanine and isoleucine. And for this one, also two carbonyls sticking up. So this is the dipeptide. There's two different amino acids we need to identify. This carbonyl, here's its alpha carbon. Go to the left, we get to a carbon. Go to the right, we get to a nitrogen. That means it's the next amino acid. Here's its carbonyl. Here's its alpha carbon. So this guy, again, this is kind of weird the way they drew it. Here's its alpha carbon. The side chain comes down. We have a CH, and then two CH3s coming off. If you look at the structures in those tables, you'll recognize this as valine. Here we talked about this one. It's just like this one. It's an alanine, CH, and then but we have this phenyl ring. So there was phenylalanine with an OH attached to it, so this is like a phenol. Actually, this is not phenylalanine, this is tyrosine. So we have tyrosine and valine. Now, I wouldn't expect you to memorize these. I'm not going to ask you to memorize them, but if you have your book in front of you, you can focus on where these alpha carbons are, find the side chain, and then just match up the side chain with those in your book, and you should be able to come up with these. The next one here, a little bit harder. What's going on here? Okay, well, here we have a carbonyl. We go here, we get to a carbon. The nitrogen's here, right? So here's the carbon. This would be the alpha carbon and an amino group. No, all right, same thing here. Here's a carbon or a carbonyl. We go to the left. Here's an alpha carbon. Attached to that alpha carbon is an amino group. Now it's not protonated. Maybe I should make this NH3 with a plus charge. So they just kind of changed the way it's drawn a little bit. The side chain is this guy here, right? The phenyl ring with the OH. That's this. So this is a tyrosine. The first one in this peptide is a tyrosine. Find the next carbonyl. Instead of, here they were both up. This one's down, but this is a carbonyl. Go to the left to get to a carbon. Here's its alpha carbon. Now you don't see a side chain here. Here's this nitrogen. Right? So this is the next amino acid. Well, the side chain isn't really missing. It's just that the side chain is a hydrogen. Right? They don't put hydrogens in here. So this is an amino acid with a hydrogen as a side chain. This is glycine. Right? The one that is not chiral, because this carbon, this alpha carbon, has two hydrogens. Fine. Next amino acid. Right, here's its alpha, or here's its carbonyl. Here's its alpha carbon. Again, you don't see a side chain. Here is the amine. So this guy is also glycine. So we have glycine, glycine, tyrosine, glycine, glycine. This funky thing, what is going on here? All right. Here you see a carbonyl going down. We go to the left, we get to a carbon. Right, that's an alpha carbon. Go to the right, get to a nitrogen. This is a different amino acid now. Okay, so what's attached to this alpha carbon? Well, we have a nitrogen, right, just as we had here. Carbonyl, go to the left, we get an alpha carbon. Carbonyl, here's this alpha carbon. Here's this nitrogen. Carbonyl, alpha carbon, nitrogen. What's attached to this alpha carbon? It's this, it would be a CH2 here, and then a phenyl ring. So this is like alanine with a phenyl. This is phenylalanine. And then the last one, alpha carbon. Whoops. The alpha carbon is here. It's attached to a nitrogen. And this is not ionized like it was here or here. This is a C-terminal. We can maybe get rid of that hydrogen and say, okay, this is a carboxylate. So this is a carboxy terminus. If this is the alpha carbon, right, we have the acid, alpha amino. We have an amino group attached to the alpha carbon. This is the side chain. The only side chain we had that had sulfur, this is a thioether, had sulfur. We had a carbon, sulfur, carbon bond. This is methionine. So methionine is abbreviated MET, met. So tyrosine, phenylalanine, methionine. Methionine is also interesting. This really kind of gets more into the chapter on DNA and whatnot when we talk about translation. So just real quick, DNA is in the nucleus, and it gets transcribed or converted to messenger RNA, and then messenger RNA is translated. So this is transcribed or transcription. Transcription is a process of copying DNA into messenger RNA. Messenger RNA leaves the nucleus, goes into the ribosomes or the rough endoplasmic reticulum. You can read the chapter on DNA if you wish. That's extra credit. I don't cover that in the lecture. But then this messenger RNA is translated, so translation into protein. Well, turns out that all proteins start off with methionine. Methionine is what we call well, they call this the start codon or whatever, but the first amino acid that starts all proteins is methionine. Later on, we'll talk about how proteins get processed so that it doesn't necessarily stay as the first amino acid, but in the translation, all proteins start with methionine. So in translation, all proteins or peptides start with methionine. Kind of an interesting fact. So it turns out that there are many biologically active peptides, or I should say polypeptides. So remember, these are polymers of amino acids that can be, well, really anywhere from 3 to maybe 50 different amino acids. So here we have these enkephalins. These are involved in painkillers. They're similar to endorphins. The endorphins contain 16 to 31, but these enkephalins, this is, I can't read that. It's things like methionine enkephalin. Well, we have a tyrosine. Here's the side chain. Here's a hydrogen, so it's glycine. Another glycine. Here's another phenylalanine, and here is methionine. So they have a methionine at the end here, so this methionine enkephalin. Here, this is leucine enkephalin. Again, it's a tyrosine, glycine, alanine, but then it's a leucine enkephalin, or leucine on the C-terminus. So again, amino terminus to C-terminus. Others, substance P. Oxytocin is interesting. Oxytocin is interesting because it actually kind of wraps around and forms this ring. So if you remember, cysteine is, let me put here, cysteine, oxytocin contains 2 different cysteines. Cysteine is the only amino acid, so I'll write AA for amino acid, that has a thiol. Remember thiols from that chapter on alcohols? Has a thiol as a side chain. This will become really important later. I'll just mention it briefly here. The thiol is an SH, and if you look back at your notes, maybe you'll see me where I had talked about how thiols can get oxidized and you form a disulfide. So this is the thiol, the side chain of one amino acid, and it reacts with the thiol of a different cysteine, and it forms a disulfide. So oxytocin, vasopressin also forms this disulfide. The difference here is highlighted. One's isoleucine, one's phenylalanine. Here it's leucine, here it's arginine. Glycine is here, the C-terminus. So it's the same C-terminus, it's just these 2 differences. Another really important one that I added here is angiotensin. And maybe we'll talk more about that later. Angiotensin, here's the sequence of angiotensin. And then there is an angiotensin converting enzyme. So I'll just do this. There's an enzyme called angiotensin. This is angiotensin 2. So it's angiotensin converting enzyme, ACE2, angiotensin converting enzyme 2, that takes this guy and lops off the C-terminal phenylalanine. So I put the 1-letter codes here. So aspartic acid is D, arginine is R, valine V, and so forth. Isoleucine I, histidine H, proline P. It breaks the peptide bond. This enzyme breaks the peptide bond here between proline and phenylalanine to produce angiotensin 1 through 7. So there's 1, 2, 3, 4, 5, 6, 7. It breaks that bond. You have angiotensin 1 through 7. Releases a free phenylalanine residue. And the enzyme here, why is this important? Well, the whole reason I shouldn't say the whole reason, but a good part of the reason I'm giving this lecture at home and not in front of you is because this ACE2 is a receptor on endothelial cells found in the lung. And the coronavirus binds to this receptor, binds to this enzyme. And maybe we can talk more about that and how the COVID disease, this acute respiratory disease promulgated by the fact that the virus binds to this ACE2 enzyme, which is on the surface of these cells. By the way, you don't have to know the names of any of these or the sequences or anything like that. All I expect you to know is that there's some biologically active peptides that are just, they're small. They're 3 to 5 to maybe 50 amino acids linked together. But they're not quite proteins yet. We'll talk more about proteins later. All right. Speaking of proteins, what roles do proteins serve? So these peptides, actually, let me back up a minute. These peptides can be neurotransmitters. They can have a hormone type function, but they're not quite proteins. Okay, they're not quite big enough to be proteins yet. So what roles do proteins serve? Well, basically, if you want anything done in a cell, you're going to have to ask a protein to do it for you. So some people make a big deal about DNA and genetics, and you hear things like, PCR, which deals with DNA, or CRISPR-Cas9, which is a gene editing procedure. So we'll just call this gene editing. Well, none of that is possible without proteins. So it's the proteins that duplicate your DNA. If you're interacting with your environment, if you're getting energy from that environment. So there's enzymes that involve, like trypsin down here, that's involved in digestion. Later on, we'll learn about other enzymes that extract energy from our food. If you need to move around, your muscles, your not only skeletal muscles but cardiac muscles, pumping your blood flow, all of that is done through proteins. With the COVID-19 pandemic, your immune response also mediated through proteins, all sorts of regulatory functions through proteins, your eyesight, nervous impulses, all that is carried out through proteins. We have peptides, let's say, or polypeptides, let's say 3 to 50 amino acid residues. Once they get bigger than 50, then they become what we would call a protein. Distinction between a polypeptide and a protein is kind of arbitrary. This is a very it's not very well defined, but let's say, greater than 50 amino acids or more. Okay, then we will say we'll start calling that a protein and not just a polypeptide. It says here fibrous versus globular proteins. Fibrous, as the name suggests, are kind of rod-like. They're rod-like. They form long rod-like proteins. Globular proteins, as the name suggests, are more spherical. It turns out that fibrous proteins tend to be insoluble, whereas globular proteins are usually soluble in water, so soluble inside the cell. Fibrous proteins, no. So proteins that tend to have a structural role or a contractile role, these tend to be fibrous. Okay. So collagen and tendons, cartilage, keratin. You don't want, wool. You don't want the sheep's hair, its wool, to dissolve when it rains, right? Or that would be really bad for your sweaters too. For contractile movement, muscles, myosin, actin fibers, right? These are also fibrous. The rest of these tend to be more or less globular, especially enzymes tend to be globular. There's some exceptions to that. Some proteins, for example, immunoglobulins, they are soluble, but we'll see the structure of them later. They might have kind of a fibrous domain and then a globular domain. So there's, again, not hard and fast rules for these as well. But most enzymes, most proteins that are enzymes, these are the ones that catalyze reactions, they tend to be globular, and those that have structural or contractile roles tend to be fibrous. Transport storage, these guys also. Hemoglobin is also globular. I'll just put here. These also tend to be globular. I'm sure there's some exceptions, and some have kind of a, like I said, it'll have a globular domain, so part of it will be globular and the other part will be fibrous. All right, so electrophoresis. This is a laboratory technique. We didn't get to do a lab involving electrophoresis, but as the name suggests, electro has something to do with electricity. I'm not sure what the name phoresis means, but basically it's a way of separating amino acids or proteins. So separation of amino acids or proteins. In the one of the labs we did, one of the labs that was scheduled, we do a separation of amino acids based on chromatography, but in electrophoresis, based more or less on the charge. So they talk about charged amino acids. Let me just write over here chromatography. Chromatography. This is also a separation technique. It's a separation, and it's based on, there's two main types. It could either be based on the size of the individual particles, so we're, the size of the polypeptide or protein, how many amino acids are in there, or it could also be based on solubility, and that's what we would have done in the laboratory, was a chromatography based on solubility. All right. Anyway, electrophoresis is, follows an electric current, so it has to do with charge. So remember, an amino acid is electrically neutral at its isoelectric point. It's pi, so it does not migrate, right? So when the pH of a solution equals the isoelectric point, the pi, the amino acid is neutral and it won't migrate. Okay? So if you have a neutral amino acid, so for example valine, and it's at its isoelectric point, it won't move, right? So this is, let's say this is at pH equals 6.0, right? That's the isoelectric point for valine. So it's neutral, it doesn't move. Now it's a Zwitter ion, right? So it has the positive on the amine and the negative on the carboxylate, but the two neutralize each other, so it doesn't move. But at a pH of 6, okay, that's fairly low, the lysine is going to be positively charged. Right? So here at a pH of 6, this is below the pi for lysine. So how do you get to a low pH? Remember that? How do you get to a low pH? I just say here low pH. How do you get to low pH? You add acid. Acid is high, has a positive charge. So the lysine will be positively charged. And that means it's going to be drawn towards the negative electrode. Right? So the lysine is positively charged, moves in the direction towards the negative charge. Opposites attract. All right, well, if this is, again, the pH here is about 6.0, this is greater than the pi of aspartic acid. I can't remember what it is for lysine. I want to say the pi is about 8, I think, for lysine. So at a pH of 6, but here, I believe the pi is about 8. I'm going to have to double check on that. But here we are at a pH of 6. That is below the pi, right? So it becomes positively charged. But aspartic acid, I believe the pI for aspartic acid is about, let's say 2 to 3. We'll just say 3 to be safe. So this here, the pH is above, right? So the pH is greater than, pH is above the pI. So it has a high pH. How do you get to a high pH? Right, you add hydroxide ions, right? Remember that? When the pH is above the isoelectric point, right, you have negative charge, right? Hydroxide ions, higher pH. So the aspartic acid here has a negative charge. That's going to be drawn towards the positive charge. Again, opposites attract. All right, so that'll do it for this lecture. The next lecture, we will finish up this chapter. We'll talk about the structure of proteins. These proteins, remember we had primary, secondary, tertiary amines, or primary, secondary, tertiary alcohols. Well, there's a primary, secondary, tertiary, and also a quaternary structure for proteins, and that's what we'll be talking about in the next lecture.